In this mitosis simulation, you're going to use pipe cleaners to represent chromosomes. Our starting materials will be 2 long sets of chromosomes, shown here in purple and blue, 2 short sets of chromosomes, shown in pink and green, and then we're going to have 4 tiny little pipe cleaners shown in red. Those are going to be the centromeres that will hold the 2 chromatids together. If you look at the procedures, we will do each step as it goes. The first procedure says that on your table to put 2 long chromosomes, one of each color. So one's from mom, one's from dad. So I've bent them just to show so that they fit in the space that I've provided for them. Also place one of each color shorter chromosome near the longer ones. So I'm going to put a pink and a green. The next procedure says to draw number 2, draw a nucleus around the chromosomes. It says to do it in chalk, but I'm going to do it on the whiteboard. This is my nucleus. Number 3, draw a cell membrane around the nucleus. So the cell membrane is going to go around the nucleus and be larger. This is your starting cell. Inside it, the next question asks how many chromosomes are in this cell. The answer is 4. There's 2 long chromosomes and 2 short chromosomes. Now you're going to fill out your paper. So on this paper we want to show our starting cell. Draw what we have. So in our box, this outer circle, that's going to be our cell membrane. So if we look here, this is the cell membrane, so we do not have to draw that. We're just interested in drawing what's here. So what do I have? I'm going to start by making the chromosomes and then drawing a nucleus around them. So I have 2 long chromosomes, I have 2 shorter chromosomes, and they are in a nucleus. This is now going to be labeled our starting cell. And this cell has a total of 4 chromosomes inside the nucleus, 2 long ones and 2 short ones. Each is a homologous pair. So this is what we're starting with. The purpose of mitosis in the cell cycle is to make a cell copy that has the same exact genetic material. So when we're done, we should have not 1 cell, but we should have 2 cells that are exactly the same. So in this next phase, the beginning of the cell cycle, we're going to start with interphase. So during this phase, one of the main things that happens is DNA is copied. So to show this, we're going to put the matching pipe cleaner. So the 2 blue pipe cleaners, 2 long blue pipe cleaners will come together, and we're going to attach them with the small red pipe cleaner. This is now the centromere that's holding the 2 chromatids together. So each of these is going to get copied. So then my purple chromosome is going to get copied. So now I have a chromosome with 2 chromatids, each is an exact copy, held together by a centromere. And finally I'm going to do the 2 smaller ones. Again, what's happening here is that DNA is making a copy of itself, so that each cell is going to get an exact copy. So the next step, it says your cell grows bigger. To make room for this process, it says to erase the cell membrane and redraw it bigger. I don't have a whole lot of space to draw it bigger, so I'm going to leave mine as is. And it says to put your 2 centrioles in one end of the cell. So these centrioles are going to set up ends of the cell. And I'm going to just move my chromosomes so they fit inside the nucleus. So what do I now have for interphase? My DNA has copied, so each of the chromosomes is an exact copy. The centrioles have shown up and the cell has gotten bigger. So now it's time to show what this is going to look like on your paper. So in the next box under the starting cell, so our starting cell had 4 chromosomes. We still have just 4 chromosomes in interphase, but they have copies, so they look like the X's. Those 4 chromosomes, each of these is still considered one chromosome because it's attached, is surrounded by a nuclear membrane, and we have 2 centrioles that have shown up. These are structures that are going to help the cell divide. So this phase is called interphase, and the 2 main things that happen is that DNA copies and the cell gets bigger. During the next phase, which is the first phase of mitosis, we have prophase. The DNA, in getting ready to divide, is going to coil. So each of these chromosomes is going to become very, very tightly coiled. So to show that, we're going to just wrap it around our finger or a pencil in order to get it coiled. So now they're going to really look like X's. They're going to take up less space and it's going to be a lot less messy. So now all my DNA is coiled. My chromosomes are coiled tightly. So the next thing that happens during prophase is, now that we've coiled the DNA, our nucleus can begin to disappear. So I'm going to show this by taking out and making the nucleus look dashed. It's starting to disappear because in order for the cell to divide the DNA between the 2 cells, there can't be a nucleus present. The last thing that happens in prophase is the centrioles move to opposite sides. So now we have sides of the cell set up and we're ready to draw prophase on your paper. So now what I'm going to show in this first phase of mitosis, prophase, what the main thing that happens is the chromosomes become visible because DNA coils up tightly and the nucleus begins to disappear. So you'll notice in the picture I still have 2 long chromosomes and 2 short chromosomes, and I've shown that they're coiled by drawing them squiggly. They also still have a nucleus, but I show that it's disappearing by making it dotted. The centrioles have moved to opposite sides of the cell. We're now ready for metaphase. During metaphase, the nucleus completely disappears. So I'm going to erase the nucleus right now. It is completely gone because the chromosomes are completely coiled up, and now they're going to get ready to divide. So the chromosomes in metaphase line up in the middle of the cell between the 2 centrioles. So they line up between them. Spindle fibers, which are protein fibers, come out of the centrioles and attach to each of the chromosomes. These spindle fibers are going to be important because what they're going to do is, in the next stage, they're going to pull our chromosomes apart. So here is metaphase. All of the chromosomes are lined up in a single file down the middle. Spindle fibers have formed and attached to each chromosome from each side. We're now going to get ready to draw this on our paper. So on our papers, the second phase of mitosis is called metaphase. The key feature of this phase is that the chromosomes line up in the middle of the cell getting ready to separate. One of the ways to remember this is M for metaphase and M for middle. You'll notice in the picture they are still coiled, so I drew them squiggly, and you can still tell that there are 2 large chromosomes and 2 small chromosomes. I am grading for the size of the chromosomes because it's important because we're making exact copies. Then you'll see that there are the two centrioles on either end of the cell. And from these centrioles we have our spindle fibers which attach to each and every chromosome. We're now ready for anaphase. After mitosis, anaphase occurs and the chromatids separate from each other. So I've already removed the centromere that holds the two together, and all that happens is that the chromosomes separate from each other. The spindle fibers pull them apart and they are now separated, and they get pulled towards the centrioles and the opposite ends of the cell. So how would you show this on your paper? So we are now on the third phase of mitosis. The name is anaphase. And the main thing that happens is in this phase, the sister chromatids separate from each other and move to opposite ends of the cell. So I've shown the chromatids separated. They're still attached to the spindle fibers. And I've still shown that I have two long chromosomes and two short chromosomes. In telophase, the final stage of mitosis, the chromosomes have now moved to the opposite end of the cell. I now have one of each color. I have a blue, a purple, a green, and a pink at each end of the cell. I'm going to move my chromosomes out of the way. The spindle fibers disappear. They've done their job and they've pulled the chromosomes apart to the end of the cell. So I'm going to place my four chromosomes, my blue, purple, green, and pink, at the end of the cell, and I'm going to draw a new nucleus. It's one of the key things that occurs in this stage is that a new nucleus forms. I'm going to do the same thing on the other side, and then the cell is going to start to pinch. So now telophase is complete. In each end of the cell, a new nucleus has formed around each set of chromosomes. In each side, I have a blue, a purple, which are my long chromosomes, a green, and a pink. So on this side, I have blue and purple, green, and pink. The cell begins to pinch because it's getting ready to now the two cells are separate. But mitosis is done because the nucleus has finished dividing. So how is this going to look on your paper? So this last and final phase of mitosis is called telophase. And the main thing that happens is that a new nuclear membrane reforms around each set of chromosomes. So in my picture, you can see that on each end of the cell, I have two long chromosomes, which are my blue and my purple, and two short chromosomes, which would be my green and pink. I still have a centriole, it hasn't gone away, in the cell, and now we're ready for the last and final stage of the cell cycle. In this stage, the cells are going to simply split apart. So the last and final stage of the cell cycle is cytokinesis. In cytokinesis, the two cells, the pinching completely separates the two cells, and we now have two cells that are exact copies of each other. So if we take a look at our last and final step, in our last step, so this final stage is called cytokinesis. The two cells pinch apart and completely separate. So each cell has a nucleus, and inside the nucleus has a pair of long chromosomes, we call those homologous chromosomes, a pair of short chromosomes. And this is the blue, purple, green, and pink. So why they're in pairs is because one came from mom, one came from dad. So for the final wrap-up, you began with just one cell. At the end, you had a total of two cells. So if I look over here, I have one cell, two cells. The starting cell had 4 chromosomes. Each ending cell also has 4 chromosomes. So again, if I look up here, 1, 2, 3, 4. 1, 2, 3, 4. You'll also notice that I also showed them large versus small. So what does that mean? The cell cycle and mitosis creates exact copies of the parent cell. And that's important because why would our cells do this? To grow, make repairs, develop, all of those things. Sometimes they just need to replace cells that are used and no longer working anymore, which would be the repair. Growth, if you're building new bones, you're building new structures, this is also going to happen. Very important process that occurs throughout your life.